Hello everyone. In this video, I'll show you an experiment. This experiment proved that when the surface area to volume ratio is smaller, that means the greater this ratio to this ratio, and how when it's smaller, that means it can't be more efficient and perform its function very well and make the diffusion process faster and exchange material faster. Look at this video now. Try to take notes. Be careful. Here they will give you the materials, the tools and materials they used in the experiment, the steps. You have to write all of these points. These are very important. Be ready now.
A good lab to do to determine why cells are small and not large is to take cubes made of agar. In them they have phenolphthalein and a little bit of sodium hydroxide. So the phenolphthalein is a pH indicator. The sodium hydroxide changes, reacts with the pH indicator, makes the cubes of agar a bright red. And basically you can cut the cubes into three sizes. You can cut them into, the larger one is a 2.5 centimeter cube. The middle one is a 1.5 centimeter cube. And the smaller one is about a centimeter size cube. You can then take these little cubes and put them into cups that are filled with dilute hydrochloric acid. You can then wait a number of minutes and you can watch how the fusion diffuses through a cell membrane. And you can use these to calculate the rate of diffusion, thereby understanding why cells are small and not large. So here we have all three cubes in cups, and inside the cups is dilute acid. The acid is interacting with the phenolphthalein to take the color out of the agar as it diffuses across the membranes. You can see the three cubes, and then you can watch the diffusion actually occur right in front of your eyes. This is at about 30 seconds after I drop the cubes into the solutions, and we'll take a look back in about a minute. About 2 minutes into the experiment, you can see that the smaller cube is, there's only a small part that is still colored, and the larger cube still has a huge mass in the center that still has the phenolphthalein indicator in it, stained pink. So you can see the fusion actually occurring much more rapidly in the smaller cube than in the larger cube. We'll revisit this in a few more minutes. At about 5 minutes into the experiment, you can see that the smaller cube, the acid has almost diffused completely through the smaller cube, whereas in the medium cube it's about a quarter of the way through, and in the large cube the acid has diffused very little. So this lab demonstrates why cells are small. The diffusion rate remains constant. So in smaller cells, solutions, or not solutions, but the diffusion of nutrients can actually occur much more rapidly. To get to all parts of a small cell, oxygen can diffuse more quickly. Carbon dioxide can get out of the cell much more quickly if it's smaller. So here are the cubes after about 6 minutes in the acid solution. In the smaller cube, you can see that the acid has diffused through most of the cell or most of the cube already, In the medium cube, the solution has diffused through about half of the cell, and in the larger cube, you can see that a majority of the unstained or the unacidified phenolphthalein agar is still pink in the center. What I'm going to do now is I'm going to take a scalpel and cut these open, and I'll show you what they look like opened up. When they're open, you can actually have students measure how far diffusion has gone. You can have students measure the volume of the cube, the surface area of the cube, and figure out the diffusion rate per millimeter by how far the acid has diffused through the cube. So here are those cubes cut in half, and you can see the larger cube still has a vast majority of it as being unstained, unaltered by the acid. The medium cube is about diffusion went about halfway through, and in the smaller cube, there's just a tiny little speck of the original color of the basic agar. Well, the clear agar is the agar that's clear has acid in it, so it has a lower pH. The agar that's in the center that stains pink has a higher pH. Remember, the agar was made with sodium hydroxide, and the pH indicator mixed with it was phenolphthalein, and we can use acid to show diffusion through cells, a small cell, a medium cell, and a large cell. In the small cell, diffusion has gone almost completely. Diffusion of the acid has gone almost completely through the cell, thereby showing that the advantage of smaller cells.
So from this video, you saw he used different agar cubes. He used different length. The first one 2.5, the second one 1.5, the third one 1 centimeter. Here, what I want from you after you watch this video or during watching this video, I want you to do like this. You have to write a lab report. Here, I want you to write here, why are cells small? What is the objective from this experiment? Why we did this experiment? To prove what? Okay? You can watch the video more than one time and find this. Then, I want you to write what are the tools, materials he used in this experiment to do it. Then, write the procedure. What did he do? What are the steps to make this experiment? Okay? Then, write the observations. The observations here, what are you going to do? You have to write what did you see. Also, you have to make some calculation to find the volume and surface area, and write where the diffusion were faster. Find the rate, don't find the rate, just he gave you the time, but here just write the volume, surface area, and put the ratio, and write what did you see, which one the diffusion was faster. Then after you finish writing the observations, write the conclusion. What did you conclude at the end after you make your calculation? You made your calculations and you saw the results. Okay, what is the conclusion? What can you conclude from all of this experiment? Okay, now if you have any question about it, about the cell size, why it's small, as you saw, the smallest one, it has the ability to make the diffusion faster. So this is the same for our cells. So our cells, they can make the exchange materials faster, okay, perform their function efficiently. Then so this is why the cells are small. Imagine we are as a human beings, we only one cell, and this cell it's very big. Do you think it can perform its function? As bacteria it's very small and can do all the life processes? For sure no, as we learned from this lesson. So the smaller the cell, the greater this ratio, which means it's gonna be what more efficient cell. Okay. Now if you have any questions you can send me your questions, and thank you for listening.